All right. So today we're going to finish up the archaea. And by the way, I'd be happy at the end of class if you want to spend a couple of minutes talking about the exam, because I know there's some concern about that. So let's start with a group of organisms that's probably related to the Nanoarchaea. It's one of or probably now a number of these little groups that may be independent phyla, maybe not, and need some resolution. But this is one I find really interesting, Nanoarchaeum equitans. This is about as close as it gets to an archaeal pathogen. It's not really a pathogen, it's a parasite. It's an ectoparasite, not an endoparasite. But it's a parasite of other archaea. And so this is Ignococcus, and this is Nanoarchaeum. This is a standard size, it's a couple of microns in diameter. These things are about half a micron in diameter, so right on the edge of your ability to see them under a microscope. When they were first described, they were thought to be blebs off of the surface of the cell. It turns out not to be the case. Now, why did they believe that? Because they amplified with ribosomal primers and they got one sequence, right? Ignococcus. These things do have ribosomal RNAs distinct from the host, but the sequences are unusual enough that our standard primers don't amplify from it. So they had to use other techniques to find out what kind of a creature it might be. It's an obligate parasite. The Ignococcus you can grow just fine. The Nanoarchaeum you cannot. You can only grow them physically attached to the host. So, for example, if you grow Ignococcus in a culture and then put a membrane in it and try to put Nanoarchaeum on the other side of the membrane, nothing happens. They won't grow. They have to be in physical contact. And the reason for this is that these organisms have thrown away the genes for almost all the things that you need to make. So they don't make vitamins. They don't make they don't make amino acids. They don't make lipids, all that kind of stuff. Essentially, all the genes are gone, except for those in information processing. Replication, DNA repair, transcription, translation, and protein modification. That's about it for the genome of this thing. It's got one of these minimal ATPases with only some of the subunits present that's probably used as a proton pump instead of an ATP synthesis gene. They don't have cytochromes or anything like that anyway. And they're probably using this to drive active transport of stuff from the host cytoplasm into themselves. And so it's probably an energy parasite. There's a protein that looks like it might be an ATP-ADP anticorn, so it's probably pumping ADP into the host and sucking ADP in. What's really interesting about the genome of this organism is that it's exactly the opposite of the sort of genome you're used to thinking about. I mean, so here's E. coli, right? It's big, it's 4,000 genes, more or less, 4 million base pairs. And it's nicely organized in operons, right? This thing isn't like that at all. The genes are scattered around the genome, what looks like haphazardly. This has a profound effect on the organism's ability to regulate genes, to turn them up and down, right? Because instead of having a promoter with a control system, you can turn the whole thing up and down. A whole metabolic pathway can be tuned easily. The genes are everywhere. And so probably this thing doesn't control its genes very much. Probably it's got a specific state that it grows at, and that's it. This disorganization is found even within single genes. What do I mean by that? There are a number of tRNA synthetase genes in this organism where the two domains of the protein are encoded in separate genes. And the protein, and those two peptides, they come together to make the functional protein. There are some tRNAs in this gene, where a piece of it's encoded there, a piece of it's encoded over here, and then the two are brought together by transplicing. So transplicing is where you put the thing together like it's got an intron, and you get splicing, but what started out as two separate RNAs instead of a single RNA. The ribosomal operons aren't encoded together. It's a disaster in terms of gene. Yeah.
So do they think it started out that way?
So this is the interesting thing about this is there's a lot of discussion kind of in the early evolution world about how much of this organization is ancestral versus something derived. In this particular case, this is a parasite, and so probably that represents I hate to call it a degeneration, but a simplification of some kind, the result of high evolutionary rates rather than anything else. And that actually, if you look at the tree, if I can go back here to the archaeal tree, compared to some of these other organisms here, this has a relatively long branch, a high evolutionary rate, and that probably is why it's pulled down in the tree. It probably is it's probably a long branch attraction. Go back to the original. This organism was isolated originally off the coast of Iceland. It has since been found in lots of other environments. Anywhere where you've got Nanoarchaea, you're likely to have organisms like this. And now that they know what kind of primers to use to find it for ribosomal amplification, they're seeing it all over the place.